Okay, so just to give the running total so far, in glycolysis we got a net of 2 ATPs, we got 2 NADH, we got 0 of those, and we didn't get any CO2. Link reaction, didn't get any ATP, we got one of these, none of these, and one of these. Now I'm going to turn this times it by 2 because we get 2 molecules of pyruvate per molecule of glucose, so we'll write per C6H12O6 because I'm too lazy to write glucose. Okay, and 2 times that. Then in the Krebs cycle, per pyruvate, we got 1 ATP, 3 NADH, 1 FADH, and 2 CO2s, and then times them all by 2. Ugh. Just do the same. So overall, per molecule of glucose, we've got 1, 2, 3, 4, 5, 6 ATPs, 1, 2, 3, 4, add 3 is 7 NADH. No, is that right? No, that's wrong. Pardon me. So you get 2 times 3, which is 6, 7, 8, 9, you have 10 NADH. I've got to times it by 2. Then you have 2 FADH, and you have times that by 2 as well. I should have written that. 4 add 2 is 6 CO2. So this is per glucose molecule. So what are we going to do with all our NADH and FADH? That's gone. That's happy. Happy doing its stuff. Okay. So the next stage is oxidative phosphorylation. Yay. And the whole point of this process is to get our ATP, because at the end of the day, all our cells want is to have a little bit of energy to go and do the cool stuff. So, if we start at the beginning, because that's always the best place to start. So we have our reduced NAD, so this has come from Mr. Krebs, Mr. Glyke, and Link. And so NADH is dehydrogenated, so we go from reduced NAD to NAD, and that means that NAD can go off and do all its cool stuff in glycolysis and the Krebs cycle, everywhere where NAD is needed. And we get out of this, we get our H+, and we get electrons. So NADH is the hydrogen carrier. It's been carrying H+ and electrons all together, which is basically hydrogen. And now we've separated it, and we've got our H+, and we've got our electrons. So the electrons, like in photosynthesis, are accepted by a high energy electron acceptor, and then they travel down the electron transport chain, which is formed of 4 complexes, 1, 2, 3, 4, plus your ATP synthase, and they get lower in energy. And as they as the electrons go down in energy, they release their energy, and that enables H+ to be transported into the intermembrane space. And it's the build-up of H+, that's the whole point of this electron transport chain. Okay, so it's from the high energy electrons losing their energy, and that energy is being used to drive H+ across, and then the chemiosmosis, so the movement of H+ down its concentration gradient through ATP synthase, is what drives this motor, which basically drives the active site of ATP synthase and allows your ADP plus PI to be converted to ATP. And that's why it's starred, because it's a great little thing. Okay, so the way that works is, so you have so this will be a really low pH because it's loads of H+, and as it diffuses down, there's a lot of debate about actually how this works. So you don't need to know it for A level, that's for sure, but some theories say that H+ binds or affects an amino acid, and that causes the formation. But your ATP synthase basically has 3 sort of active states. So one of them, it will bind ADP plus PI, another one it will bind ATP, and the other one it will release it, and it just goes round cycling the formation of ATP from ADP plus PI. And you get other ATP powered sort of proton pumps, which is sort of a similar thing, just going in the reverse. But anyway, your H+, which has gone down here and then it's come back round again, forming our nice ATP. It's joined with the electrons over here, binds to oxygen, which is the final electron acceptor. Always a mark in exams, often a mark in exams, and you get your H2O. So yeah. There's also debate about how many ATPs you get per H+, but I think it's supposed to be around 30 per molecule of glucose. Okay, oh yeah, and FADH. So FADH doesn't actually lose its hydrogen to this lot, but it just adds more electrons, so you get a greater electron reduction, and that happens on this second complex. And you also have this last bit, it is called cytochrome oxidase, and that's what reduces your oxygen to H2O, and cyanide binds there and inhibits this process, and that's why cyanide kills you pretty damn quickly. Okay.